Hello and welcome to Biology Practical 1 with myself and obviously Mr. Taki, and this is about microscopy. Now, as you can see from Mr. Taki right here, what he's taken is the outer layer, or the epidermis, of the onion itself. Now, why he's doing this is so we can see right through it, so we haven't got layers upon layers of cells. Now, once he's placed it on the slide, hopefully without putting his fingers on it, okay, we will add a drop of methyl blue. Now this is to dye the cells themselves so they can be seen more easily under a microscope. Now we place the cover slip on just to keep all the dye and everything in place. Okay. All right, and it's ready to go under the microscope. Now, once it's under the microscope, we can start to see the cells themselves and get an idea of the size and magnitude of them.
Right, just to add to what Sir has stated there, you start with a magnification of 10, okay? There are various parts of the microscope. This is the eyepiece. These are the objective lenses. You have one that says ×4, which is a magnification of 4, and then you have X10, and you also have, for some microscopes, you have up to 100, but you also have 40 as well. So you need to make sure it is at 10, and then you turn on the lights on your microscope, and you use the coarse adjustment, this one, to move the slide very close to your objective lens. Okay? Look through to see whether you can see the image. Just move the coarse adjustment up and down until your image appears under your microscope. Then you can use a fine adjustment, this one, to make the image more detailed. The moment the image is very detailed, you can use that to move the stage until you can clearly see the cells under the microscope.
Okay.
Go on, sir.
Now, in an exam sort of situation, obviously you wouldn't be able to do this. What they would ask you is more of the magnification type questions. Now, magnification is quite easy to work out. What we do is we take the image size, which will be given to you in exam, or you'll have to measure it, divided it by the actual size of the image. Now, this can be quite easily shown in the triangle below, with magnification and image actual size on the bottom and image size above. Now, one thing that a lot of people try to struggle with is changing units. So micrometers is by far the most common for measuring the size of cells, and millimeters and meters is again more of the image size. So to go from micrometers to millimeters, we times it by 1,000. From millimeters to meters, times that by 1,000 again.
The other thing I want to highlight as well is that this practical, they can ask you how to actually carry out the experiment, not just to interpret the results at the end of the experiment. So they can actually ask you, how do you prepare a slide to observe under the microscope? Then you can describe the stages which I've explained. And they can also give you a slide that is already prepared, an image, and then you are asked to calculate the size of that image, or the actual image, or the image size, and then the magnification as well. Remember the difference between magnification and resolution. Magnification makes the image larger. Resolution makes the image more detailed. And other things you should know around this is about the difference between light and electron microscopes. Light microscopes, they have a lower magnification compared to electron microscopes. Electron microscopes have a far higher magnification, but they only look at dead samples. Thank you for watching the video, and I hope this is helpful.